I think this is probably a good time to go ahead and get started. So I'm delighted to be here with everyone tonight. I am Dr. Chris Pettit. I'm the Division Chief of Pediatric Cardiology here at Columbia University. It's a real honor to kick off this research salon on behalf of Dr. Kimara Targoff, who you all know. And I'm going to start by introducing Dr. Jordan Orange, who is the chair of the department here at Columbia. And he will say a few words about just the exciting work and really the vision for Columbia University and pediatrics writ large. Thank you, Jordan.
Yeah, no, thanks, Chris. And it's so great to be with everyone this evening. And it's so great to be supporting Dr. Targoff this evening as well. And by the end of the evening, that image that you see on your screen will make perfect sense. And you'll know how that image is actually going to transform the health of children. And that's what we do. That's what we do here. That's what's happened here for almost 150 years. Innovation, advancement of the future of children, and creation of the future for children. This is the third oldest children's hospital in the United States, staffed by our faculty, our teams, and a consistent series of innovation throughout its long history. We're one of the top pediatric programs in the country. US News and World Report just the other week rated us the top pediatric program in the metropolitan area, tri-state region. But that's not because we're running ads on TV or on the back of taxis. That's because people like Dr. Targoff do phenomenal work day in, day out, and are internationally recognized for innovating, making a difference. And our faculty are the people that people want to come train with, want to learn from, and quite frankly, follow day in, day out. That said, I really look forward to the future of her research program. It is unlike, and you'll hear more from Dr. Pettit, unlike just about any world over. And why we pulled together this salon series, of which this is one of the beginnings, is to really help our extraordinary leaders here on the faculty at Columbia connect with the city in which we exist. And really be able to grow in what our city appreciates is happening here, and that our city can feel part of it and advance it, because the work that happens here really can transform, and I believe will transform. And with that, I certainly couldn't be more excited that we have Chris Pettit here at Columbia. Many of you know and have been long-standing supporters of cardiology. The cardiology division at Columbia for children has always been one of the very best in the world, tracing back to so many firsts. First heart transplant, the first device put in a 2-day-old to support heart circulation. And again, having some of the very best internationally at what can be done in heart health for children. Of course, I'm pleased that we're going to see a massive expansion of that in the next phase of growth of what we do here. And a key part of that is Chris Pettit coming here to really begin to lead the next chapter of that. Chris is the division chief of cardiology. He's an internationally renowned cardiac interventionalist. And is already growing teams and programs. And I'm so pleased that he is an ardent supporter of Dr. Targoff in her laboratory. But Chris, we're so grateful that you moved here from Atlanta Children's Hospital and have really hit the ground running, and so grateful to be partnered with you as we support the extraordinary program of Dr. Targoff. So Chris, thank you for inviting me. Kimara, it's so great to see you, and I'm so glad you're sharing your research with the New York City community.
Thank you so much.
Dr. Orange, thank you for that really lovely overview and really call to action for all of us. And it's an honor to be part of such a fantastic team. My job tonight is actually really easy because I don't have to explain slides like what Dr. Targoff is about to explain. As Dr. Orange said, I'm a clinician scientist, and I take care of infants and children and adults who are born with heart disease. But my job tonight is really to just briefly give you some sort of a sense of context of pediatric heart disease. So, children born with heart disease, you think of this as a very tragic and very sad event that happens, but actually it turns out that congenital heart disease or heart defects are the most common birth defect that children and infants are born with, which is a little bit surprising. About 1% of all babies born have some form of heart defect. Some of those are mild and some of those are quite complex. And actually the heart forms, the earliest organ to form in the fetus is the heart. Which is basically mostly done with its evolution and complete formation by 42 days post-conception. So there's some really important things going on even before a mother is aware she's pregnant that have a huge impact on the life of the child and the course of that family. When these children are born, they come to a center like Columbia, and they oftentimes need some sort of intervention in their early lifetime, whether they're a newborn baby or an infant or an adolescent. And here at Columbia, we are one of the busiest centers in the world, both with heart transplant and heart failure, but also with really innovative therapies, whether it be in the operating room for complex surgical repairs or in the cath lab for device closure of defects. So I'm really proud of our team here. As Dr. Orange said, our legacy here at Columbia is enriched by our innovation, both clinical innovation at the bedside, but also in the research forum. And we have a whole host of exciting researchers here within the Heart Center who are working on understanding the best way to treat children with heart disease, the best therapies, the best operations, and the impact on the social and neurodevelopmental outcomes of these children, and the impact on the family as well. So a lot of exciting research on that front is going on. But Dr. Targoff works on something completely unique and something which is really critical to our understanding of heart disease. And that is, how does it form and what is it that causes heart disease to develop in the first place? And I'm really excited to have her discuss this with you in a way that I think will be really engaging and really exciting. So Dr. Targoff, I'm going to hand it over to you now.
Okay, great. Thank you so much, Dr. Orange and Dr. Pettit. It's wonderful to be here, and I really appreciate the very kind introduction, and I'm very excited to share all my work today with everyone. So the goal of my talk today is to really tell you about how understanding the genetics of cardiac development and regeneration are necessary to improve human health. And just to give you a quick background, I think you kind of heard it a little bit from the others, but my background is as a clinician, as a pediatric cardiologist. And with this clinical training, I became very interested in trying to get at the underlying basis for why we have defects in heart development. And so in order to do that, I focused in on some basic science research and embryology, and then ultimately genetics. And I'm gonna tell you a little bit about that storyline as we move forward. So I'm gonna start off now by describing a quote from Ralph Waldo Emerson: "All life is an experiment," and that is definitely how I've treated my life, and it's how I've treated my academic career as well too. I have focused primarily on clinical care to start with, and trying to improve the health of patients in general, pediatric patients specifically. And then ultimately knowing that many of the defects that we see come from early origins, as Dr. Pettit just mentioned, during fetal life and embryogenesis, and how this kind of multi-system organ developmental processes occur and integrate with each other. But given our focus as a pediatric cardiology department division, we are particularly interested in understanding how the heart forms. And what that means is how the tissues come together and become interacting, aligned, function properly. And then in order to get at that, we knew there are some guiding factors, and those guiding factors are really the underlying genetics. And so it's been shown clearly in much literature that basically genetics guide the formation and the movements and the migrations of different cell populations in the embryo that help to make this heart a beautifully contractile and life-supporting organ. And what I'm going to tell you today is basically how all of these questions in this real cycle of experimental design and inquiries really can be answered by looking at the genetic regulation of cardiac development in zebrafish. And I will elucidate why this model system is really ideal to help us investigate our questions further. So, So in terms of, I think you got enough of an introduction on the Division of Pediatric Cardiology at Columbia University Irving Medical Center, but again, just want to hone in on where we are. This is an excellent, outstanding, preeminent institution in the country and in the world, primarily for pediatrics and also for pediatric cardiology. The innovation and the firsts, as both Drs. Pettit and Orange mentioned, are really quite amazing at this institution. And I was trained here as a resident, as a fellow, and have embodied this commitment really for about 20 years now. So I actually, since you did hear a lot about the clinical mission here at the Division of Pediatric Cardiology, I want to dive really into why we are studying. Sorry, my pointer is a little bit finicky. While we're studying congenital heart disease. So for anyone who's not totally clear about congenital heart disease, it really means any sort of defect that occurs during the formation of the heart, and usually with which patients are born. As you can see here, the heart, it has 4 chambers, 2 ventricle and 2 atria. And as we know, it's the most common birth defect in humans, and patients who are born with congenital heart disease actually do have a lot of challenges and require medical interventions, surgical interventions. And there's been significant advancement in these interventions such that we're able to take care of patients in such a successful manner that greater than 90% of our patients actually survive to adulthood. However, much of this investment has come through research and the need to improve our care and our understanding of the basic underlying causes of these disease processes. And so that's where my commitment comes in. And actually, I'll just interject. I am not going to speak for too long, about 20 minutes or so at the most, but then there'll definitely be room for questions. So please hold on to your questions because I would love to have a dynamic conversation. Apologize for not mentioning that sooner. In order to understand congenital heart disease, as I mentioned, we have to look early in development. And so heart development happens within the first 40 to 50 days of life in the human. And ultimately, to get to this large, like functional biventricular biatrial heart, we start early in embryogenesis with just 2 little strips of cardiac cells, what we call progenitors that basically grow to become the heart. And they start right here at day 15. You barely know you're pregnant. They merge to form what we call as a linear heart tube, and then loop appropriately to have blood flow in this direction and out that direction through the aorta to the whole entire heart. So my question is, how does each cell know what to do? How do they migrate together? How do they extend, form, loop, and become this beautiful, again, very powerful heart that beats for our entire life? And in order to do that, we've kind of cycled backwards to get at a more basic cellular and even genetic level. And it's the genes that truly guide these processes. And so that's where we get at the concept of the genetics of congenital heart disease. What genes lead to these processes? And it's not just one gene, one problem. It's a very complex multifactorial problem. So for a quick review for anyone who's not totally remembering all about genes from high school or maybe college, we have the nice coding elements of DNA that's basically the alphabet to tell us what genes make what proteins. It's transcribed to RNA and then ultimately translated to proteins. And proteins are the functional units within the cell that help tell the cell what to do. And I know many of you heard about RNA recently in the past few years, but we've been studying it for years now, and many labs have been doing so as well. So at this moment, there's greater than 400 genes in which there are variants that are associated with congenital heart disease. But still, in about 50% of patients, the underlying cause is unknown. So clearly, there's still a lot of work yet to be done. And the real advancements in These inquiries will be gained from understanding the kind of integration of genomic data as I'm outlining here. This is what I mean, understanding of sequencing and of DNA, RNA, and proteins, and combining that with what I showed you on the previous slide of developmental biology, how the heart forms. And merging these concepts is really what we do in our lab. And ultimately, this will help us to answer how the variability in genetic defects are associated with different outcomes in patients with congenital heart disease, and will help us to guide clinical care. Okay, so now we say, well, how are you going to do all this, right? And as I mentioned before, I'm going to answer, not I and many team members and many scientists as well are eager to answer this question with the zebrafish. The zebrafish is an ideal model system for the study of congenital heart disease. And there's many reasons why. And so I'll try to kind of lay that out very clearly. First and foremost, there is a real similarity between the human and the zebrafish genome. There's about 70% of the same genes. The genome is completely sequenced. We know every single nucleotide, and we're able to look at this, all the different genes and areas between coding genes as well too, which is quite important. But very importantly, and this is different from, you might say, why don't we look in mouse or rats or other kind of mammalian models, mammalian meaning like, the baby's born in the uterus, and as are humans. But the reason we don't do that is because while there are many assets to those models, the reason we focus on this model is because it really offers an advantage in terms of the transparency. As you can see here, this is the eye of the zebrafish embryo, the mouth, this is the whole heart right here, and the yolk sac and the extension. And this is the atrium with the heart muscle cells here, the blood vessels here, the blood cells here, and this is the ventricle as well. So it's only one chamber for each in the zebrafish, but it's, again, very similar morphologically and also in terms of genetically. And so because we can see it, we can interact with this embryo and we can adjust things genetically. We can alter how we look at different cell types. We can look at the atrium, we can look at the ventricle with different, what we call transgenes, genes that will light up with fluorescent proteins, different cell types. So there's so much we can do. And lastly, and very importantly, and this is what's unique to the zebrafish above and beyond like any other model systems, is that the cardiovascular system, which is normally needed to pump oxygen to the entire body and have the embryo live, is not required for oxygenation, because you can have diffusion of the oxygen through this very thin-skinned embryo. And in doing so, we can study defects in the heart that would otherwise kill any other embryo or mouse or so forth. And so we can have the living organism alive, follow what happens when the heart becomes damaged, and then understand those processes. And that is really powerful. So, okay, just to give you a little sense of one other kind of beautiful aspect of the zebrafish embryonic development is really to see how quickly it happens. So it's as I mentioned earlier, and Dr. Pettit mentioned as well, the human heart and mouse heart develops over the course of, like, let's say 30, 40, 50 days. But in about 2 days, we have a functional zebrafish heart. So this is hours post-fertilization. And as soon as this egg is fertilized, and that's external fertilization, we have one cell here with the yolk, it's fed off the yolk. And in the course of 4 hours, you have about 1,000 cells. And then basically by the course of 1 day, this is an embryo with a head, an eye, the brain, and the trunk. And then there's a little heart tube that starts right around here. And by 2 days, you have functional heart. So this allows us to do high throughput genetic screens, to look at many different genes, to have hundreds of embryos, not, again, like a normal litter for any sort of other, humans or mice or other animals are much smaller. So this is a powerful genetic tool. So now I'm just going to show you a little bit about where we get these fish, where we keep them, how we manage them. And so I'm going to start, I'll show you the volume for 1 second, and then I'll cut out. I just want you to hear kind of the beautiful water and the dynamic nature of this facility. So here, this is our zebrafish facility. And we have over about 200,000 fish in this facility. And as you can see here, I'm panning over to show you the life support systems, which are the multiple UV filters and water filters. These are the shrimp brine that we hatch every single day to feed our zebrafish. And these are the nursery area where our babies are kept. So these are 1 week old zebrafish embryos, then they grow to about a month old and move to another section where they're fed differently and nurtured in order to grow as quickly as possible. And then we go around to the adult side that you're seeing here, where they're all adult zebrafish, and we can then mate them. And what you see is in a mating cage, put together a male and a female, and overnight, basically, they lay anywhere from 100 to 200 embryos. And so it's a very humane, comfortable, and useful way to target embryonic development and to understand these processes. And as you can see here, again, we have so many different fish with so many different genetic mutations and also transgenes, as I mentioned. So these are all different labels with myriad different tools that we can have. And these fish, as you can see, grow nicely for about 2 years and provide us with a real asset, a very strong basis from which to do our work. And so now just going to the next phase of what do we do with these embryos? We actually image them. We take a look at the heart live, and this is a collaboration that I've been working on with Dr. Elizabeth Hillman in the biomedical engineering department here at Columbia University. And that's one other thing that the university offers, is an extensive scientific community that has absolutely enhanced the program that we run here in the Targoff Lab. And this is a microscope that she designed called SCAPE, which is allows to see live cells interaction in a volumetric, meaning like you're taking the whole volume at a time, as opposed to like one slice and then putting it together. And she and I together published in a high profile paper in Nature Methods in 2019, the first application of this microscope to zebrafish cardiac development. And what you're seeing here, these are the labels of transgenes for anyone who's a little bit more genetically inclined, and they label red blood cells and endothelium, that's the inner lining of the heart. And this is the inside of the heart labeled in green, and the red blood cells in red flowing into the atrium, through the valve, into the ventricle, and then out the outflow tract to the rest of the body to perfuse the rest of the body and provide oxygen. So really with these tools, as you can see, we can look at individual cells. We can genetically manipulate individual cells, see how they interact with their environment, with the tissue as a whole, with the organ as a whole, and can deduce so many different can draw so many different conclusions about how genes regulate form and function. So, okay, who's doing all this work? Well, in our lab of cardiac development and regeneration, we have a really wonderful team, an outstanding team. This is a picture of some members, not everyone's included, and this is a list of some members who've been really quite instrumental in the past years. And we have members of our lab ranging from postdoctoral fellows who have completed their PhD to graduate students and master's students, and also undergraduates. And once again, I really do want to emphasize twofold. Being at Columbia University really has an amazing resource in terms of educationally to be able to draw together trainees from such different levels of education and provide them with experiences that will help them grow into active and productive physician scientists is really an honor, I have to say. And in addition to my clinical role and my scientific role, as you can see here, one very prominent mission in my life and within the life here at Columbia is to obviously educate. And I spend a lot of my time working with fellows and residents and graduate students and helping them train, helping them think scientifically, and helping them grow as best they can. So that is a true commitment in our lab, and we hope to continue with that mission going forward with many new trainees down the pipeline. So to introduce you to a little bit of these people, I want to kind of take you back from the fish facility where we get our embryos and we bring them back to our lab space. We do our molecular biology, our basic lab experiments. This is Dr. Alleman. I'm sorry, not doctor. She's a graduate student, Gelly Alleman, and she is in the Department of Physiology and Cellular Biophysics, and she's doing her PhD in our lab. And this is Caitlin Ford, another graduate student from the Department of Genetics and Development, who's working on a very interesting project looking at the development of head muscles and cardiac development together, as many different patients have connected abnormalities in these features. This is Nicola, who is a postdoctoral fellow, and he, as you can see here, is looking in on the zebrafish embryos in the dish and collecting them and separating them according to different features that they have. So this is what we work, this is what we do. And so now I'm just going to kind of bring you to what we found. I'll basically summarize about, many years worth of work in about 2 to 3 slides. So don't worry, it's not going to be overly burdensome, but I'm hoping it will provide you with some insight as to the discoveries that we've made and how we hope to apply those going forward. So we focused in our lab on NKX 2.5. It is a gene that's called a transcription factor. A transcription factor is important to regulate other genes. So it's kind of like up high in the hierarchy. And this transcription factor, the reason I focused on this years ago was because it was the first and really the most predominant gene associated with congenital heart disease. And so our lab was the first lab to develop a zebrafish, to generate a zebrafish mutant in NKX 2.5. And so we were really excited to uncover some key roles of NKX 2.5, a whole bunch of them actually, that we've published on serially over the past couple years. And so just to clarify here what we're looking at, this is the whole wild type or normal zebrafish embryo, and the NKX 2.5 mutant has abnormalities within the propagation of the blood. You can see collection or backlog of the blood. So what that means is the heart is not functioning properly. And when you get down into the more high-powered view of the heart, you see here a nice normal ventricle, and this ventricle in the mutant is small and not pumping the blood well. So it's accumulating more in the atrial chamber. And so you can also see that here in another image where we use antibody stains to look at, the atrium and the ventricle and the very small ventricle that we see. So once again, the NKX 2.5 is required to make the ventricular chamber form properly. And the ventricular chamber is what pumps the blood through the entire heart, through the entire body. So we've extended this work to include many different tissue types, cell types, and what I would say temporal resolution, meaning timing. So in development, everyone thinks about, we all think about where things happen and when they happen, right? So what gene is operating in what cell at what point? And so we've kind of dissected out these pathways and have really illuminated many different genetic networks guiding heart development, regulated by NKX 2.5 in the outflow region of the heart, in the inflow region of the heart. These genes operate there, and this also are important for developing the conduction system of the heart, and then also within the atrium and the ventricle. And finally, in terms of other functions on an embryonic level of NKX 2.5, is we focused on the electrical conduction system. So in addition to forming the heart function and the heart shape, basically, also, as I think many of you probably know from family members who have different conduction abnormalities, such as atrial fibrillation, when the heart doesn't beat normally, it's very uncomfortable and it doesn't function normally. And so we know that NKX 2.5 is important to establish that normal conduction within the heart. It is here in the wild type versus the mutant. Differences in the heat map pattern where you can see the conduction is proceeding very nicely over the wild type heart and kind of in a much slower fashion over the mutant heart. So we are also extending these studies to share our thoughts with collaborators. And I have really been excited by some new directions in the lab, focusing on what's called extracellular matrix, which is the tissue surrounding the heart cells, and have collaborated with a series of biomedical engineers, as I mentioned, Dr. Hillman early on. Another collaborator currently is Dr. Vidula, and he focuses on modeling cardiac mechanics and blood flow. And so we've worked together now in the zebrafish to take images of the zebrafish, extract these images, and this is what Dr. Vidula has done, and actually use the images to reconstruct the movement of the heart. And if you just look closely at these movies, you know, all the undulations of the different tissue are so essential to help the blood flow properly, to help the pressure and help the volume relationships of the different chambers. And then he's able to extend these studies to look at how the blood cells move and what flow is. And flow of the blood is important for pressure within the heart. And so this is mimicking the blood flow into the heart and then out of the heart. And then this one is really quite the most essential movie, as you can see here, because the red is highlighting where there's high pressure moments within the heart in red. And that's important both for the development of the heart. You need pressure to help the heart form, and also bad pressure that can develop secondary to, let's say, surgical repair or just congenital heart disease in general, can also cause problems within the heart too. So therefore, these tools will help us to elucidate how cardiac mechanics, flow, and pressure really alter how the heart forms properly, abnormally, and what we can do to fix that. So, okay, now I'm going to change directions a little bit. And I do see some questions building up, and I will definitely get to all those. We will definitely get to all those shortly. So the next kind of little storyline that I want to tell you, again briefly, is about cardiac regeneration and how cardiac regeneration is important to highlight the many different cardiac regeneration is used to basically treat failure within the heart cells, right? And so specifically, zebrafish is a great model for this because we actually see a really positive response to injury. And when I say injury, like ischemia, heart attack, things like that. So you see in the zebrafish, we're actually able to cut off the ventricular chamber and the whole zebrafish ventricle can grow. And in the neonatal mouse, which is like one week old mouse, there is a scar that forms when you cut off blood supply, but it is somewhat improved. So there is some regenerative potential. But in the adult mouse, equivalent to the humans, you actually have a complete scar formation, which leads to dysfunction with the heart movement and basically leads to heart failure. And I'm sure, again, many of you may know family or friends that do have these issues. And so our question is, what is it about these early developmental stages that allow the mouse to regenerate? And so that's where we bring in our developmental genetics, our NKX 2.5 knowledge, and trying to understand maybe how that plays a role and looking at it in zebrafish, because zebrafish is able to regenerate. And so from a evolutionary biology perspective, you can imagine that there actually are embryonic kind of tools within the heart that exist within the fish that, as you've gotten higher up the ladder, have been suppressed. And I can talk about many different reasons for why they've been suppressed. It can have to do with temperature regulation, outside, needs for temperature regulation, needs for thyroid hormone, many different reasons, but those regenerative potentials have been suppressed. So now we need to learn how to harness that. So I want to just to kind of regroup a little bit on what I'm talking about clinically. So regeneration is important for the treatment of ischemic heart disease. And ischemic heart disease is basically the worldwide leading cause of death. And as you can see, this is like the heart from the outside. Before I showed you a cross section, these are the ventricular chambers, and this is the coronary arteries. And when you get a blockage in the coronary artery, which is obviously interior, you get subsequent loss of cardiac muscle, and that leads to scar tissue and impaired contractility. And we don't have good tools to treat these patients. Really, they're treated medically. And in very bad cases, they're treated with cardiac transplant, which, as you can imagine, is not an ideal outcome. And obviously, and there are also challenges within organ donor limitations and so forth. So really, to able to enhance cardiac regenerative potential within the heart muscle itself is kind of the holy grail. And so we are eager to use our knowledge from zebrafish to make this happen. And some of the techniques that have been discussed and reported more recently as being potentially successful are the injection of new cells into the heart. And more excitingly, from our perspective, is the idea of heart cell proliferation or cardiac regeneration through proliferative mechanisms, right? So basically, this is a little picture to show us a heart cell, and this implies that it's cycling and proliferating. So our goal is to get these cells that have been damaged to kind of get to a state where they can actually regrow again and replenish that area. So I'm going to tell you how we do that. We have a very interesting model that's actually was not designed in our lab, a colleague of mine many years ago, but we have pioneered it here at Columbia in our lab recently using a ventricular resection technique. We also use a cryoprobe too to induce injury with a little gold probe. But basically, we can cut open the zebrafish chest, expose the ventricle, excise off the ventricular tissue, and there, as you can kind of see here through this little figure, that there's quick clotting, and with the clotting, there is it permits survival. So the zebrafish does not bleed out as you would if you did that in a mouse or a human, of course. So we can do this, and then we can observe regeneration initiate within hours, and then occur over the course of the next 2 months. And so I guess I'll just share again our data and our findings in a quick 2 slides, but to years of work. But here we've shown very excitingly, as I kind of note down here, this is a prior postdoc from the lab who's now gone on to study, to pursue her scientific career in Portugal. And so we have in press, in Nature Communications, again, a very exciting journal to identify basically our findings, describing how NKX 2.5 is necessary for cardiac regenerative potential. Again, a developmental gene is required in the adult heart to heal the heart. And this is basically the experimental design. We have the early embryos that we raise to adulthood. We perform the resection, and then we examine them after 30 days. And we look at the wild type and the mutant. And what you can see is the heart muscle here is not completely, but looks nice and normal and doesn't have the more pinkish hue of the fibrin or the collagen. But here in the mutant, it's very densely fibrinous and collagenous, and has significant scar tissue, which it 100% impairs the cardiac contractility. So this was a very exciting finding, and ultimately we imagine we could actually resupply NKX 2.5 to the heart, to an injured heart in essence. And we're eager to extend these studies and to test them in mouse and humans as well. And before I move on to the next slide, I want to just highlight, so we also did what's called RNA sequencing, and this is like genetic or genomic studies to understand all the genes that are to regulate this process. And so again, in our recent publication, we've shown that NKX 2.5 governs networks and networks of different genetic interactions. And all you can see here, the genes that are listed here, which I know it's a kind of overwhelming slide, but I just wanted to show you the ones that highlighted with blue and pink are the center foci with which NKX interacts to mediate all these different genetic processes. So that is our recent discovery, and we are eager to move forward and implement these studies going forward with additional genes and uncover different mechanisms that may ultimately help us to understand how to treat heart disease even in a more improved fashion. So just to kind of summarize at this point, I want to say how our studies have shown excitingly that NKX 2.5 is essential for cardiac development and regeneration. We use a zebrafish embryo to look at embryonic heart formation, function, and electrical conduction. We also use the adult zebrafish to look at different ways in which cells can proliferate, as you can see here, and what's called dedifferentiate, which basically means you take a cell that has already a nice functional adult cardiomyocyte, you can make it go back to a little bit more of an embryonic state, kind of like a stem cell, right? I know many of you probably have heard of stem cells, embryonic state, and then basically it can regrow and then come back again to a more differentiated or more adult-like form to function in an effective process in the adult zebrafish or the adult human as well. So these studies are summarized in some of our literature here. It's not a comprehensive list that we published basically consecutively over the years and tried to kind of grow our research program in new directions and ultimately solve questions that we continue to ask ourselves that will translate back to our understanding of congenital heart disease and ischemic heart disease as well too. So again, what can we do with all this? I just want to emphasize that the applications to cardiac development and regeneration research are critical. We use this knowledge of NKH 2.5 mediating heart formation and function, and we can ultimately look at different ways to predict surgical outcomes. We can identify associated congenital defects and know how to predict that early on in utero when the baby's obviously not born, and can help provide counsel for and prenatal care appropriately, and also ultimately to enhance survival of our patients as well. And in the adult heart, we're eager to use both proliferation and dedifferentiation mediated by NKX 2.5 to help alleviate many of the defects that are associated with heart dysfunction and myocardial ischemia. So as I mentioned, we want to unleash and open up these developmental mechanisms that are native to the zebrafish and also exist in the mouse heart, but need to be kind of de-repressed. And then we replenish cardiac cells following injury, and ultimately we're looking to improve cardiac function with these therapeutic interventions. So while we have gone so far and we've really grown the lab and our research program significantly, we've made really instrumental and innovative discoveries. There's always more to do, and there are many areas in which we are eager to invest in order to grow our lab and grow our mission. And as I mentioned, the important focus on education and training of graduate students, on the development of technologies and innovation in order to, let's say for us, work with the zebrafish, image the zebrafish, do modeling within the zebrafish. And this, for example, is the hand-built microscope scape that I mentioned by Dr. Hillman, which clearly requires a whole team, screw by screw and test by test, to really figure out exactly the nuances of this, microscopic optics and physics modules that are put together in order to image the volumetrically moving heart that we mentioned before. And lastly, the development of interdisciplinary projects. So with Dr. Vijay Vendula, we are very excited to look at these, the function of the heart, the flow of the heart, and truly how this creates stress on the heart wall, both to enhance growth and also for potential deterioration of muscle in pathological circumstances. So basically, with all these efforts, we're excited to continue our mission and move forward and really make additional advances, as this is something I'm obviously so committed to and am exhilarated to be a part of all the time. So with that, I really do want to thank everyone here. First and foremost, thank you for your attention and your time. Dr. Orange as the chair of pediatrics, and Dr. Pettit as chief of cardiology. And all my collaborators in the School of Engineering, and our bioinformatics collaborator who happens to be in Australia. And very importantly, also to thank my funding mechanisms from the NIH and the multiple grants that I've obtained from that funding source in a very supportive fashion, and also other New York State stem cell initiatives and Congenital Heart Disease Coalition. So thank you so much, and I'm eager to hear any questions that you may have.
Dr. Targoff, that was really inspirational, and congratulations for all the work that you've done in this space, and for being really phenomenal as a investigator and as someone who runs a lab, which is really the source of so many discoveries. So thank you for being extraordinary and for mentoring and sort of developing the next sort of generation of scientists as well. I'm going to take a step back before we get into the questions, and we'll be done by 7, I promise. I had the privilege about 1.5 years ago to participate in sort of refereeing your Targoff's promotion. I was just starting at Columbia, and what was really great about that was that I had the opportunity to see how the international community who is working on cardiac development, how they view your work, and world leaders are universally just in awe of the work that comes out of your lab, in awe of you, and are really inspired by you. Some of the audience tonight should understand that some of the techniques that you described, that you demonstrated so eloquently tonight, those were developed by you and have really changed how research in this field is done. So congratulations on that. It's not lost on me as the division chief that most heart centers for pediatrics in the United States do not have someone like you. You're a real treasure. The fact that you were able to attain RO1 funding from the NIH, the National Institute of Health, that is really, not only is that vital to your work in terms of funding, but it's also clearly a reflection of what they believe the impact of your work is. So congratulations there. I wanted to say finally, before we get into questions, that sort of the support for Dr. Targoff's lab, both in financial resources and sort of in other ways, really goes toward Columbia's vision and mission of being excellent and being the top heart center really in the country. And that's what drives clinicians and researchers like Dr. Targoff, like myself, to come to work every day, is really to create the #1 heart center for children in the country. So with that, maybe we should jump into some of the questions. And I've been looking through them during your presentation. I think there's a few themes, so some of them are thematically similar, but maybe I can start off, Dr. Targoff, by asking you, are there other zebrafish researchers, and why and how do you work together? Do you collaborate together on sort of on your work, or are you the only one doing zebrafish research?
Oh, no. I'm far from the only one doing zebrafish. I don't want to give anyone that impression. I'd say no. It's a relatively small community scientifically. I'll try to answer it. So actually, we feel very proud of our zebrafish community in the sense that we kind of, in a little nerdy way, joke about the lineage, which is like, the genetic lineage, because it really started in about 1998. And where the first zebrafish lab began, and it was in Tübingen and then in Mass General as well. And from those labs, literally everyone it's like my grandfather, for example, like that's who started in the lab, and for me it was in Tübingen. And so that community is very close, is an international community, and we work really well together, and there's a lot of collaboration. So, but to what Chris was saying, I actually am the first zebrafish investigator here at Columbia, and with the support of the institution at the time, and when I was kind of hired back, we built the zebrafish community. So that has been wonderful. And not until 2019 did I have actually one colleague who's joined. So it's really one other person and myself here. So yes, it's a little lonely here, but internationally and globally, I have many friends, and we're going, many colleagues to work with and to learn from, 100%.
That's fantastic. Another question I thought was really interesting is what's the biggest near-term human problem your research might help solve or cure?
It's a good question, very good question. And I have to say, I do tend to think that the regeneration work will be really instrumental, because with this insights into specifically NKX 2.5 and different genes that are guiding cardiac, the ability to proliferate heart cells that have otherwise been thought of as non-proliferative. can be paradigm shifting. Like it will really, and so actually, as I say that, so we can do that in the adult heart where there is injury due to, let's say, the heart attack, the ischemia concept, but actually it's also can be really applied quite in the near term to the embryonic heart when there is loss of tissue. Like many of our, as again, Dr. Pettit referred to and so forth, many of our patients with congenital heart disease have hypoplasia or small ventricular chambers. And that's one of the most lethal types of diseases we have. And if in utero, we could actually induce on some level the growth of those cells, that would be game changing in terms of life expectancy and outcomes for individual patients. And this type of concept is not necessarily so far away. I don't think it's tomorrow, but it's within our lifespan.
That's phenomenal. I like this question a lot. With 400 genes involved in congenital heart disease, how do you choose which is the most important?
So that's a very good question. And, you know, it's, I think it's a probably simple and a complex way to look at that. I would say I'm not really trying to study only the genes that are identified. We're actually trying to identify new genes. So it's not as if I'm looking at that list of 400. I did focus initially on this NKX 2.5 because it is a master regulator. And I think that understanding that role is essential. But really what we're looking at now is, as I mentioned, RNA sequencing and genomics research. So we're taking all these deep sequencing and high throughput sequencing studies to actually look at all the other genes that are regulated. And so the goal is to get kind of these networks and these pathways and to understand how they're all working together. So it's not like, let me take a gene that's known and study it. It's actually a little bit the contrary. Let me get new genes, help to understand in my model, and then go back to humans and see how that may be altering their outcomes clinically.
That's fantastic. Thank you. There are many questions on the theme of funding. And so many people here, and I'm really gratified to see these questions come in about what type of support do you need? What is the most impactful way to support your work and the work of the mentees who work with you?
Yeah, well, thank you very much for that. And I appreciate that for all, anyone who's asked that question. It's, as much support as we get here at Columbia, as much support as I have been able to obtain from the NIH, it is constantly a challenge. And it's a challenge because there are just limitations with what one gets normally in terms of what you can do to expand your ideas and expand your research. So really, I think it's kind of what I was showing on one of those last slides, like it's the idea to support a trainee, like a salary support for a trainee. Honestly, that's huge. And it's not necessarily a huge amount of money, but to have another person, another mind that's going to be there with you and work, that sometimes funds are the limiting factor for that. And a lot of it just has to do with the nature of funding and the nature of grants and grant writing. And so that's one key thing. And then I guess the other arena is really is technology. And we spend, I'll just throw it out there. For example, there's I didn't show these data today, but I wrote a grant just recently that I submitted to the NIH in March, really to look at what's called like tissue elasticity. So it's actually more of like a biomechanics perspective, and this is with another collaborator. We want to look at how this outflow tract moves and how it's elastic and how it kind of absorbs energy, and as a way to understand outflow tract defects in humans. And what we need to do is get a little probe connected. It's like a catheterization. I haven't even talked to Dr. Pettit about it. Catheterization to probe into the zebrafish outflow tract to get pressure measurements in order to use pressure and movement in order to deduce elasticity. And so like that tech, that is commercially available, but I think it's about like $20,000 to $30,000 itself, right? So these are limiting factors. So we have not purchased that yet. Hopefully we'll get the grant funding. So, we have abundance of ideas, and the implementation takes resources.
I can also provide some context. I'm not a basic science researcher, so I'm a little bit out of my element here, but my observation has been that, funding from the federal government and from large organizations kind of comes in these waves. It's kind of feast or famine, and many successful basic science labs have a bedrock of sort of philanthropy, which sort of supports between these sort of ebbs and flows of the grant funding cycles. And so I think that's sort of some of the opportunity I think we have tonight, is to sort of provide that sort of foundation that continues the important work that you do. Obviously, I will say that I know that you are a very enthusiastic and very talented grant writer, and I know that your NIH grant funding will continue, but this is an opportunity to sort of provide that bedrock, is kind of how I see it.
100%. Yeah. Thank you for that.
Yeah. I think we have time for one more question before we wrap it up. This has really been wonderful. And I really appreciate such a lovely discussion tonight from you, Dr. Targoff. The last question is, I really like this one too. What do you love most about being a pediatric cardiologist?
What do I love most about being a pediatric cardiologist? Well, I guess it's really why I went into it to start with is, I mean, it's a sequential thought process, but the first one is I loved working with kids, and that's why I went into pediatrics. I really just couldn't imagine working with adults. No offense to everyone here, but kids are just precious, right? And then within cardiology, I think that I remember doing a report like in 8th grade on like how the heart forms and how that functions. It just always fascinated me. It's the physiology, it's the anatomy, and how it integrates. And it's very three-dimensional. Like, most tissues are not that way. And this is an organ that just works in such a kind of like beautiful, as I said, undulating movement type oriented fashion. And to really be able to get at those kind of physical properties and fluid properties and anatomical elements are it's exciting to me. So that is how I think about kind of like the disease process, and then the patients themselves. I feel the other element is I loved pediatric cardiology as opposed to, let's say, other pediatric subspecialties, because we were able to do something. And that was really hugely instrumental. And like most of what we do, a lot of it is surgical, right? And then we take care of the patients, but we can fix them, and we can make them better. We can make them have a whole life ahead of them, and then they can have babies. And, we're taking care of those patients now too. So that is something that to go from such a sick child to a really, at least almost healthy adult is a beautiful thing. So whatever I can do to contribute to that metamorphosis, I should say, I'm happy to do so, both clinically and scientifically as well.
That's very well said. I echo those sentiments completely. Yeah. Well, it's now 7 o'clock. We've really hit it perfectly in terms of time. I will just sign off by saying thank you to everybody for your time tonight. Thank you again to Dr. Targoff for a great presentation. And we will have as you sign off tonight, you'll see a way to contribute. And I know that Allison and the rest of the team can also provide a mechanism for people to provide support for your important work, Dr. Targoff. Thank you again, everybody.
Thank you, everyone. And thank you, Dr. Pettit. Bye-bye.
Good night.
Good night.